So, I should hit record. Am I recording? I hope I'm recording. I am recording. Okay, good. Let's talk about the nervous system. Guys, gals, our last unit of the semester is going to be nothing but the nervous system. Most students, including myself when I was in your shoes, find the nervous system to be the single most difficult body system out of every body system that's covered. This is the hurdle. If we can make it past this unit, everything in Bio 314 is going to seem easier to most of you. And that's just the general pattern. The nervous system is very complicated. And the thing is with the nervous system is that every year, to do it right, we'd have to add another 20 or 30 pages to the textbook, because every year there's that many new discoveries being made right now about our nervous system. The idea, there's lots of big picture questions like, where is your consciousness in your brain? Where's your personality? What makes you you? What is physically, what's a memory? What's the anatomy of memory recall? There are lots of big picture questions right now that we're just starting to scratch the surface on. So guys, gals, I want to encourage you to hunker down. I know we're at the home stretch. Summer vacation is looming for most of us. But we have to finish strong. Don't slack off. You should be studying consistently every night to be successful with the nervous system. To reiterate, as we look at our nervous system, guys, gals, the nervous system and the endocrine system are used for communication. When we think of the endocrine system, we think of slow communication with chemicals in the bloodstream. When we think of the nervous system, it's going to be fast, quick, electrically based communication using our nerves, or excuse me, our neurons to direct information from one part of the body to another part of the body. And when we think of our nervous system, there's just kind of a general broad overview. First, information goes in, and this information going in is typically going to be picked up by some kind of a sense organ. We can have touch, we can have temperature, we can have smell, taste, vision, hearing. There's all kinds of sensory modalities or ways to have that information entering our body. That information enters our body, and all of these different signals need to be turned into electric signals. So we have to take a smell, and we can take two different smells, and we have to turn them into two different electric signals that can be processed by the brain. We have to take two different noises and a taste, and we have to process all these different sensations and turn them into different kinds of electric impulses to be processed by the brain. And when we think of the brain and the spinal cord, it's the hallmark, it's the middle, it's the central part of our nervous system. It's referred to as the CNS, our central nervous system. This is a big, broad umbrella term that includes the brain and the spinal cord. So this information is sent to the spinal cord and the brain. Some of it's processed in the spinal cord. Most of it's going to be processed in the brain. The vast majority will be processed in the brain. And then a decision is made, and an action is decided, and then the command is issued. And the brain and or spinal cord will send a signal out, usually to some kind of a muscle, occasionally to a neuroendocrine gland, and then action is taken. So in a broad sense, a really big picture sense, we take that signal, turn it to electricity, send it to the brain, process it, and then send an electric response away from the brain. That's kind of a good summary of the entire nervous system. But we're going to talk about more details. Don't worry. Now, when we think of the brain, that's the central nervous system. The PNS, the peripheral nervous system, is everything outside of the brain and spinal cord that involves nervous tissue. So when we think of our peripheral nervous system, we're going to think of nerves and ganglion. And we should also throw in there sensory receptors as well. Depending on how you slice and dice it, sometimes you can consider a sensory receptor part of a nerve, sometimes it's not considered part of a nerve. So everything outside of the brain and spinal cord that's involved with the nervous system is the peripheral nervous system, the PNS. Now when we think of a nerve, I really need to emphasize this for you, because nerve and neuron are two concepts, two ideas that students will often get flip-flopped. This is one of those classic student misconceptions. A nerve is not the same thing as a neuron. A neuron is not the same thing as a nerve. They're related but different. A neuron is a specific cell. A nerve is a bundle of axons from many different neurons. And another name for the axon of a neuron is a nerve fiber. So when you think of skeletal muscle gross anatomy, how we have the bundles of bundles of bundles with the epimysium, the perimysium wrapped around fascicles, and then the endomysium. When we look at the nerve anatomy, we have the epineuronium, the perineuronium, and then the endoneuronium, all wrapped around these bundles of bundles. And the individual fibers in a nerve, instead of being a skeletal muscle cell, are going to be individual axons from individual neurons. And when we think of a ganglion in the peripheral nervous system, I want you to think of the cell bodies of neurons. Cell bodies of neurons can group together and form a bulge. This bulge in the peripheral nervous system is referred to as a ganglion. This is something you should commit to memory. You need to be able to draw this on command. That's how well you need to commit this to memory. This is our roadmap. It's our guidebook to the nervous system. So when we look at our nervous system, there's two big umbrella terms, the central nervous system and the peripheral nervous system. Our central nervous system is divided into the brain and the spinal cord. The peripheral nervous system is going to have subdivisions, and these subdivisions are based on what they specifically do. There's a whole chapter dedicated to these last two subdivisions. With the visceral motor division and the sympathetic and parasympathetic divisions. We'll talk about that, I believe, in 2 weeks when we get to that chapter. To broadly kind of look at these, we're going to have our sensory division send information to the central nervous system. The central nervous system makes a decision, and then that response is sent to the body through the motor division. And when we look at the motor division, we have two branches to our motor division. One we have control over, conscious control, it's voluntary control. That's known as the somatic motor division. So when we think of somatic motor division, typically we're going to be focusing on skeletal muscle tissue because you choose almost always when you're contracting your skeletal muscle tissues. There's a few exceptions. And then when we think of the visceral motor division, I want you to think of cardiac muscle tissue, glandular secretions, and smooth muscle tissue. So these are going to be the involuntary signals that your brain sends out to your body. So as we look at the peripheral nervous system, that sensory division can also be referred to as the afferent division. Afferent's more of an old school term. I'm a big fan of calling it the sensory division because it's your senses. It's the information, and that's just, to me, it's an easier to remember name. But afferent is still pretty common in the literature, and depending on the text you're looking at, you'll see that term. So here's how you can think of the afferent. Here's a word analogy for you. Afferent accesses the brain. Information will access via the afferent division. A and A, afferent accesses. So information is going to be sent to the central nervous system via the afferent division of the peripheral nervous system. And within that afferent division, there's two subdivisions. We're going to have somatic sensory information. And this somatic sensory information is going to be from our skin, our bones, our muscles, our joints, and then we're going to have that visceral sensory division. And that's going to focus on information being carried from the organs, typically the abdominal pelvic cavity. When we think of special senses like sight, sound, hearing, those are typically going to be categorized in the somatic sensory division. As a caveat, depending on the textbook you look at, whether it's a Saladin versus a Meriden Home versus a Kensington, there's different big textbooks in A&P. These subdivisions of the nervous system will vary a little bit from textbook to textbook because there's a lot of debate right now. This is a hot button field of research. we're going to stick with your lecture textbook for this class. We also have the division of the peripheral nervous system that sends information away, the motor or efferent division. And when we think of the efferent division, to remember that word, I want you to think of the signal exiting via the efferent division. Exit efferent. So the afferent information accesses the central nervous system. The efferent, with an E, exits the central nervous system. And within that efferent motor division, we have the stuff that happens automatically and the stuff that we have conscious control over. That stuff that happens automatically is often referred to as the autonomic nervous system because it happens automatically without conscious control. So with that visceral motor division, think of the organs of your abdominal, pelvic, and thoracic cavities. You don't choose when your intestines contract. You don't choose when your heart contracts. These are things that all just happen automatically. They're part of the visceral motor division or autonomic motor system. And then we're going to have the somatic motor division. Our somatic motor division is going to focus on skeletal muscle tissues, and it's going to be voluntary in nature. So when you choose to run, when you choose to move, that particular signal is going to be sent out of your central nervous system through the somatic division of the efferent division. And then within the autonomic nervous system, there are two subdivisions of the autonomic nervous system. We have the sympathetic and we have the parasympathetic. And this is one of those situations where there's really a lot of deviation from textbook to textbook. Some textbooks will throw a third subdivision within the autonomic nervous system. Your textbook has just the classic two, sympathetic and parasympathetic. So that's the way we're going to go with this division. So when we think of sympathetic, I want you to think of high stress situations. A sympathetic nervous system is going to help you with your flight, or freeze response. When you're scared, when you're fighting, or when you're running away, your sympathetic nervous system is activated. The word analogy I like to think of is sympathetic stress. The stressful sympathetic. When you're agitated, it's the sympathetic nervous system. And then we have the parasympathetic nervous system. Our parasympathetic nervous system is classically referred to as the rest and digest branch of the autonomic nervous system. So when we think of everything associated with resting and digesting, we're going to focus on the parasympathetic division of the autonomic nervous system. So when we activate the parasympathetic nervous system, blood pressure tends to go down. We tend to preferentially shuttle more blood to the digestive tract. We tend to feel feelings of wooziness, and there's a give and take. Our sympathetic division helps us to make it through that stressful environment and survive through that stressful environment. And it's good to be alive, awake, alert, enthusiastic when you have your sympathetic nervous system activated. But you can't be stressed out all the time. You need time for tissue repair. You need time to absorb nutrients. You need time to sleep and cycle waste products out of your central nervous system while you're sleeping. And this is where the parasympathetic nervous system comes in. We need both functioning on a daily basis for health, for optimal health. So let's have a review question. The blank division tends to prepare the body for action. We'll say physical action. We have the sensory afferent, motor afferent, somatic motor, parasympathetic, and sympathetic divisions. Based on how quickly the answers are flying in, I think we have a lot of confidence on this one. We're down to about 30 seconds. We have about 10 seconds. Make sure to get your answers in. Please don't wait until the last second to click submit. You want your answers registered. Click something now. All right. The correct answer was sympathetic, and we nailed it. Good work, guys. Well, I should say the best answer is sympathetic. So let's talk about neurons. And before we talk about the properties of neurons, I have an analogy for you. I love 80s rock. And John Bon Jovi is one of the best 80s rock bands in existence. But when you think of John Bon Jovi, you know, everybody talks about John Bon Jovi, but they don't talk about the light technicians, they don't talk about the sound technicians, they don't talk about the stage setup team, they don't talk about the coach bus drivers. John Bon Jovi would be a lousy musician if he didn't have dozens of people helping him be amazing. And the same is true with the nervous system. We have, you know, a lot of segregation. Everybody focuses on the neuron this, neuron that. They get a lot of attention on the neuron. But a neuron can't do its job without its support team. And yes, neurons are important, just like John Bon Jovi is awesome. But we also need to acknowledge the support team. So we're going to spend some time talking about the neurons, kind of the rock star of the nervous system, but there's those support cells that we also need to talk about, the neuroglia. So we're going to talk about both of those in the next several slides. Let's first talk about the neurons. One of the properties of a neuron is that it's irritable. It's not that it's in a bad mood, but it's that it can respond to stimulation. And as it's excitable or capable of responding to that stimulation, it typically is going to send some kind of an electric signal. Some action potential will be conducted across the membrane of that neuron. And when we think of neurons, they can also secrete neurotransmitters. And these neurotransmitters allow for that signal to go across a physical gap. Because when we think of our neurons, and if we're conducting electricity across the neuron, we need to have a continuous membrane in order for that electric signal, that electric current, to continue moving. But the moment the membrane ends, the electric signal ends, and we need to switch from electricity to a chemical messenger to get that information across the gap. And that's where the secretion of the neurotransmitters comes into play. Let's pause for a moment. Let's go back to that last slide. I should really emphasize with the conductivity, one of the things that makes neurons really unique and different from muscle tissues or muscle cells is that neurons can make and receive electric signals. Muscle cells can only receive. They do not initiate an electric signal. And as we're looking at our neurons, there's different ways we can categorize those neurons based on what they do. We call those the functional classes. Some neurons are going to focus on taking some kind of modality, sight, smell, vision, hearing, touch, temperature. And they'll take that sensation and turn it into an action potential and send it to the central nervous system. We call those neurons associated with sensation the sensory neurons. And a sensory neuron will pick up a signal, send it to the brain and spinal cord, and within the brain and spinal cord, we have a ton, and just a ginormous number of neurons. And these neurons that exclusively exist within the central nervous system serve to process information, to make decisions. We call these the interneurons. They're in between the sensation and the decision. They are the decision makers. And then after those interneurons of the central nervous system make their decision, they are going to send some kind of a signal, either through the autonomic or through the somatic peripheral nervous system divisions to make a response, to take an action. And those neurons that send the information away from the brain and spinal cord are referred to as efferent or motor neurons. So we can look at the neurons and categorize them based on what they do. We should also spend some time talking about the parts of the neuron before we talk about another way to categorize them. We can also categorize neurons based on their structure, but we can't talk about that system until we know about the structure of the neuron. So the most important part of the neuron is referred to as the neurosoma. The neurosoma sometimes has three synonyms, or there's two other synonyms I want you to commit to memory. Neurosoma is the same thing as saying cell body of neuron. They are complete and total synonyms with each other. Neurosoma means cell body of neuron, and it also is sometimes going to be referred to as the soma. Sometimes saying neuro is too much work, so we drop that prefix and just say soma. I personally am most comfortable with saying cell body of neuron. I like smaller syllables, or fewer syllables in my words as opposed to neurosoma. But all these terms mean the same thing. Now within the neurosoma, we're going to have a fairly large nucleus. And this nucleus is going to, contain the genetic information and help control the processes of our cell body. And then within the neurosoma, there are some other structures we need to highlight. We have the cytoplasm of the neuron. And within that cytoplasm, we have some inclusions. Those inclusions are going to be storage molecules. When we think of these inclusions, our neurosoma, our cell body of our neuron, needs to store some materials inside of its cell body. And it uses these raw materials to synthesize neurotransmitters. Our neurons, in addition to having to pump out tons of ATP to maintain their ion gradients for action potential purposes, are also processing tons and tons of protein molecules because they're secreting tons and tons of neurotransmitters. Now, as we look at the cytoskeleton of a neuron, the cytoskeleton of the neuron is going to originate in the cytoplasm, but then it'll extend the length of all of the branches. There's the short branches and the long branches, the dendrites and the axons. And our cytoskeleton is going to move throughout all of these branches. In particular, we're going to have a lot of cytoskeleton present within the axon. And the reason for that is that we have transport vesicles that carry neurotransmitters from the neurosoma down to the axon terminal where they can be exocytized to send the signal on. Think of how we talked about acetylcholine being released from the axon terminal at the neuromuscular junction. Those storage vesicles filled with acetylcholine had to get there somehow. They were sent there via the cytoskeleton of the cytoplasm. And when we think of a neuron, because the neurons are constantly synthesizing all of these different molecules that are being secreted, all of these neurotransmitters, they're going to have a lot of endoplasmic reticulum with a lot of ribosomes attached to it. And the endoplasmic reticulum has so many ribosomes attached to it, you can see it at 400 times magnification under a regular light microscope. It's actually a lab objective. You need to be able to recognize a Nissl body. So from a lab perspective, when we're looking at the neurosoma, the neuron, and the nucleus, and at this point in time, you can find a nucleus of the cell body of a neuron, no problem. But then there's these dark regions within the cell body. Those dark regions are the Nissl bodies, and they correspond to an endoplasmic reticulum that's working overtime to make chemicals that are being secreted. Now, we also have these branches, or these extensions that come off of the neurons, the dendrites and the axons. And there's different ways we can categorize them. So let's just talk about some general guidelines. Generally speaking, dendrites send information to the cell body of the neuron. As a rule of thumb, dendrites receive electric signals and carry the signal to the neurosoma. And then at the neurosoma, these different signals, some of them are excitatory, some of them are inhibitory. All of these different signals converge, they're processed, and then sometimes the axon will carry a signal away. Sometimes the axon does not carry the signal away. Dendrites carry information to the cell body. Axons carry information away from the cell body. So functionally, the direction of information flow is a key difference between axons and dendrites. And when we think of our axons and our dendrites, another key difference is that our neurons will typically all have one axon. But some neurons will have no dendrites, some neurons will have one or two, some neurons have dozens of dendrites. So there's a lot of variability in the number of dendrites present on a neuron. But as a rule of thumb, with very, very few exceptions, neurons will have one axon. Now, as we look at this axon, or this nerve fiber that's leading away from the neuron, these axons can be really long. It's not unusual to have an axon be 15 centimeters in length. So when we think of that in proportion to the size of the neurosoma, we can have something, a cell body of a neuron, that's about 100 microns, or 1/10 of a millimeter in diameter. And then it can have this long spindly fiber 10 centimeters long coming away from it. And we can take all of those little spindly fibers, those axons, and bundle them together to make the nerves. Now, as we're looking at the axon does have a little bit of branching that occurs. We call those branches of the axon an axon collateral. And they allow for one electric signal to be sent to multiple cells by one neuron. We have lots and lots of axon collaterals, typically at the terminal end or at the very end. These collaterals, or this branching pattern at the end, is your classic dendritic pattern. And when we think of a dendritic pattern, I don't mean dendrite. I'm talking about branches of a tree. Or for those of you who have spent any amount of time in ecology, if you look at streams going to rivers, going to major tributaries, there's that classic dendritic pattern that forms as well. Now, as we look at our axons, some axons are going to have some insulation on them called myelin. Some axons don't have the myelination. And that's going to vary from neuron to neuron. Some neurons are going to have a bunch of myelin on their axon. Some neurons don't have that myelin on their axon. And because we're going to spend a lot of time talking about the axon and how the axon sends electricity, we're going to focus on the cell membrane of the axon, the axolemma. Kind of like how the sarcolemma was the cell membrane of a muscle cell, the axolemma is the cell membrane of an axon. And within the axolemma, we have the axoplasm, kind of like the sarcoplasm of the muscle cell. And as we're looking at our neurons, at the very end of those axon collaterals, or those branches of the axon, we're going to have the axon terminal. And that axon terminal is going to be the delivery site. All of those transport vesicles that contain those neurotransmitters that were made at the Nissl bodies are going to be bundled up and then carried down the cytoskeleton and delivered to the axon terminal. And then at the axon terminal, if it becomes electrified, it will release the neurotransmitters, and those neurotransmitters will carry the information across the synapse or across the space. So we talked about the structure of the neuron. Let's talk about structural classifications of neurons. So we have functional classifications. We also have structural classifications. We can classify them based on the number of branches coming off of the cell body of the neuron, or the soma of the neuron. A unipolar neuron is going to have a single process leading away from it. And that single process is going to be referred to as an axon, but it's also going to be dendrites merged with an axon. The unipolar neurons are typically going to be associated with sensory cells or sensory organs within our skin, and we're also going to have a lot of processing interneurons within the spinal cord that will fall into this unipolar neuron classification. We also have anaxonic neurons. These are the most recently discovered neurons. Anaxonic neurons do not have axons. So these anaxonic neurons will receive lots of signals, and signals will converge at the anaxonic neurons. And they've been located particularly within the retina. So when we get to special senses, the very last week of lecture, we talked about sight and sound and equilibrium. We'll talk about these anaxonic neurons in more detail. We also have multipolar neurons. These are by far the most common kind of neuron. Overwhelmingly, the vast majority of neurons in our bodies are going to have multiple dendrites and one axon. They have multiple poles. And when we think of these multipolar neurons, one of the wonderful things about these multipolar neurons is that because they can make so many connections, they're so good at networking. They're really good at processing information. I know we don't have a lot of computer science majors in here, but let's talk a little bit about information processing within a computer. Right now, nearly every computer in existence is stuck with binary code, zeros or one. And when you look at binary at a physical level, the 0 and a 1 is an oxidized versus a reduced state of that transistor. Did it gain an electron? Did it lose an electron? One of the guys I was doing undergraduate research with was really interested in making a 3-oxidated state transistor, but that research has become obsolete. The golden, the golden child right now, or the holy grail of computing is quantum computing, where you can have instead of a transistor, you have a qubit that can exist at an infinite number of states at the same time. And you get lots more processing power out of these qubits because they can exist at many different states. When we think of these multipolar neurons, having many connections means that they can exist at many different membrane potential voltages. We can have that -70, we could have it +20, we could have -60, we could have -66. There's all these different ways to process that information. Some neurons are going to carry dendrites will carry excitatory signals that raise the voltage. Some dendrites will carry inhibitory signals that lower the membrane voltage. And all of those different signals, whether they raise the voltage or lower the voltage, will converge at the neurosoma, the cell body of the neuron, to be processed. The more connections, the more processing power. So when we think of this in terms of processing power, it makes sense that we have lots and lots of these multipolar neurons in our brain, where we do most of our information processing. We also have a lot of bipolar neurons. Bipolar neurons are going to be categorized as having an axon and a dendrite. And we have lots of bipolar neurons associated with special senses. So think of our nose, think of equilibrium in the inner ear, and think of sight. And we have another review question. What is by far the most common type of neuron? This is that neuron that's associated with lots of processing power. We're down to about 30 seconds. We have about 10 seconds. Make sure you get your answers in. Looks like we have 3 lingering responses. Oh, they're almost all in. The correct answer is multipolar, and as a class, we did phenomenal. Good work. So let's talk about how we move things in our axons. Within the axons of our neurons, we have the cytoskeleton. And that cytoskeleton can function as a 2-lane highway or a bidirectional road that can send transport vesicles. And in addition to having that cytoskeleton for the transport vesicles, we're also going to have lots of voltage-gated ion channels. In particular, voltage-gated sodium, voltage-gated potassium. Those are the 2 that get the most attention. But we also have voltage-gated chloride channels as well. So as we're looking at the axon, there's two directions we can send stuff within the axon. We can send stuff to the cell body of the neuron, and we can send stuff to the axon terminal. The movement of something to the axon terminal is referred to as anterograde transport. And then, after we deliver that neurotransmitter or transport vesicle to the axon terminal, we go through the anterograde transport. The neurotransmitters are secreted into the synapse. They're used up, and then they're chopped up by those enzymes. We talked about acetylcholine esterase within the muscular system. And after those neurotransmitters are chopped up into little pieces, the pieces are scooped up, selectively reabsorbed, packaged, and then sent back to the Nissl bodies to be recycled. And this process of sending these materials back to the neurosoma is referred to as retrograde transport. And during this transport, we have two different proteins that will carry the molecules. We have dynein and kinesin. When we think of kinesin, this is going to be a protein associated with anterograde transport. Kinesin is the protein that carries these vesicles away from the cell body to the axon terminal. And then we have dynein, and dynein is going to be the protein that carries these transport vesicles back to the cell body. And the reason we have two different proteins that go in two different directions down this filament is because there's some bidirectionality to the filament. It's not, as you look at our actin filament, the actin filament that makes up this part of the cytoskeleton is not the same. It's kind of like reading information from left to right versus right to left. Depending on the direction you move your eyeballs across the page, it'll look different. So, as we're moving things through the axon, we can move them quickly. And this, when we try to move something quickly, keep in mind, we're trying to move it a couple centimeters. 10 centimeters is not uncommon to go from the cell body to the axon terminal. So if we're moving these chemicals, and they can move between 20 and 40 millimeters per day, that would be 0.2 to 40 centimeters within a day. So what are some of the things that we're going to transmit to the axon terminal quickly? We're going to send some organelles. We'll send digestive enzymes, synaptic vesicles, and small molecules. And when we think small molecules, I want you to think neurotransmitters. And then after the axon terminal has done its thing, it's released the neurotransmitters. They're chopped up by the digestive enzymes, repackaged, recycled, and then sent back to the cell body. And when we look at this process of axonal transport and we think about disease states, we have some diseases that are associated with moving down the axon. If you've ever known somebody, it happens, if you had chicken pox, I'm having a brain hiccup right now. It's the reemergence of chicken pox after, there we go, shingles, thank you. So when we think of shingles, shingles will be associated with the path that the axons travel in our bodies because of that pathogen moving through the axon. Question, comment?
It says rate of 20 to 400. You said 20 to 40.
Did I say 20 to 40? I meant to say 400. Millimeters, 40 centimeters. Most people can think in centimeters. Most of you don't think in millimeters. Let's talk slow axonal transport. So the fast axonal transport will typically be centimeters per day. Slow axonal transport is maybe up to a centimeter per day. So we'll have 1/2 of a millimeter per day up to 10 millimeters per day. And with this slow transport is always going to be one direction. It's always going to go from the neurosoma to the axon terminal. And it's primarily going to be powered by just simple diffusion through the cytoplasm. So our fast axonal transport is going to depend on those two motor proteins we talked about, burning through ATP to haul that transport vesicle very quickly. The slow axonal transport is a passive process that doesn't require the input of ATP, but as a passive process With simple diffusion, it just takes longer. If you tear a nerve fiber, so you have some kind of an injury to your tissues, we find that this slow axonal transport is the limiting factor for repair to that damaged axon. So, we spent some time talking about the neurons. Let's talk about the neuroglia, the rocks or the support staff of the nervous system. We find, as a rule of thumb, that there's at least 10 neuroglia, 10 support cells for every one neuron. So if we have approximately 1 trillion neurons in our nervous system, we're going to have approximately 10 trillion support cells, quite a few. And our neuroglia do wildly different things. They have wildly different structures and wildly different functions associated with those different structures. So as we look at our neuroglia, they can help to serve just generally to protect the nervous system neurons, physically by forming barriers. Sometimes they maintain homeostasis for them by controlling the external environment, the cerebral spinal fluid around those neurons. If we look at the developmental process, the neuroglia direct the movement of neurons. As neurons are forming new axons, and as new axons are moving throughout the body and we're making new nerves, the neuroglia will direct the generation of the nerves while we're in utero. And as we're looking at a mature neuron, our mature neuron can't have synaptic contact with another neuron unless it has some support cells, unless it has some neuroglia helping it out. John Bon Jovi can't sing unless he has his support team. Our neurons can't send and transmit those electric signals unless they have the neuroglia helping them out. Now, within our different divisions, our central nervous system and our peripheral nervous system, there are support cells or neuroglia associated with each. The central nervous system with the brain and spinal cord is the most important, the most complex, and will have the most neuroglia. The first one we're going to talk about is the oligodendrocyte. It's kind of a fun word to say. Almost up there with sternocleidomastoid. And when we think of the oligodendrocytes, these are going to be neuroglia of the central nervous system that will wrap myelin around multiple axons of multiple neurons. And as we look at the figure that I included from your textbook, the oligodendrocyte is right here, and this one oligodendrocyte will have multiple branches going to multiple axons, and each branch will myelinate a segment of the axon. A common student misconception is that one oligodendrocyte will myelinate an entire axon. That is not true. One oligodendrocyte will myelinate part of multiple axons, or segments of separate axons. The next neuroglia are the ependymal and the microglial cells. When we think of the ependymal cells, I want you to think of the cells that are going to be associated with making and circulating cerebral spinal fluid. So our ependymal cells are going to make the liquid that we use to deliver nutrients. The cerebral spinal fluid is the fluid that gives the raw nutrients to the cells of the nervous system, particularly the central nervous system. This is going to be a simple cuboidal epithelium that is also highly ciliated. And because we have all these cilia on the ependymal cells, the cilia will beat in unison and circulate cerebral spinal fluid through the ventricles of the brain, around the outside of the brain, and then around the outside of the spinal cord. When we think of the microglia are the white blood cells of the central nervous system. We have a blood-brain barrier in place that makes it really hard for cells to go between the central nervous system and the rest of the body. So because it's really hard to get a cell across that barrier, we need to have our own native population of white blood cells within the central nervous system. And for us, that is going to be the microglia. And then we have the astrocytes. The astrocytes are so named because they look a little bit like a star. They have many, many projections coming off of them. And the astrocytes do a ton of different things. They're the the jack of all trades or the handyman of the central nervous system. And because they do so many different things, we're going to need a lot of them because of all the different functions they carry out. So let's talk about some of the highlights of the astrocytes. The most significant thing I want to emphasize to you is the astrocytes make the blood-brain barrier. Astrocytes will have extensions coming off of them that encapsulate blood capillaries, and these extensions cause the blood capillaries to develop tight junctions. And those tight junctions make it so that bacteria and other pathogens have an incredibly hard time getting into the central nervous system. It's a barrier that helps protect the central nervous system from invaders that may be in the rest of the body. But our astrocytes do more than just make the blood-brain barrier. They also maintain homeostasis for the cerebral spinal fluid. The ependymal cells make the cerebral spinal fluid. The astrocytes will tweak the chemistry or alter the chemistry of the cerebral spinal fluid. And they alter the chemistry by primarily regulating ion flow. Do we need to add a couple ions? Do we need to subtract a couple ions? Do we need a little bit more sugar? Do we need a little bit less sugar? This is where the astrocytes are going to be at play. One notable function of the astrocytes is that they help to regulate glucose concentrations. The neurons are kind of, the divas of the nervous system. They want glucose and typically only glucose. Everything else is not good enough for a neuron. So the astrocytes are going to accommodate, and they'll take lactate and turn it into glucose so that the neurons can have that constant supply of glucose that they need. And we're going to finish neuroglia of the PNS, and then we'll call it a day. So we have 2 more slides. So as we look at the neuroglia of the PNS, or the peripheral nervous system, we have the Schwann cell. The Schwann cell is going to be analogous to the oligodendrocyte. So the oligodendrocyte in the central nervous system myelinated axons. Schwann cells also myelinate axons. But there's a key difference. One oligodendrocyte will interact with multiple axons. Schwann cells will only affect one axon per one Schwann cell as it myelinates the axon. And these Schwann cells also help to guide the regeneration of the axon. If we have a damage to the axon, the Schwann cells will serve as the guide while the axon regrows. And we don't have astrocytes and a blood-brain barrier in the peripheral nervous system, so we have an ad hoc solution made with satellite cells. And these satellite cells will wrap themselves around the cell body of a neuron in the peripheral nervous system to protect that neuron from the external environment. And this will be our stopping point for the day.